everyone. Welcome to yet another episode of Health Discourse with Sakshi and Sushant. So, hi Sushant, good morning.
Good morning.
So, folks, from today onwards, we will be doing some separate discussions about cancers. I think most of us who have been following us, most of you who have seen our content, who have been following us on LinkedIn or maybe on Spotify and other podcasting platforms, I'm pretty sure you know that we are going to be coming up with a cancer detection platform soon in the month of August. So, from today onwards, in our following discussions and talks, we will be covering the pathways which impact the onset and progression of oral cancer. So, Sushant, I think let us begin this discussion by enlightening the audience about how oral cancer occurs and what are the pathways that will impact the detection. Over to you,
Okay.
Sushant.
Okay, thank you. So we just few statistics before we start deep diving into the biological mechanism which we are decoding, right? So oral cancer globally affects around 300,000 people annually every year, right? Uh, and accounts for 145,000 deaths every year, right? And the surprising fact is the oral cancer is and head and neck cancer. India leads the pack. One third of the global incidents are Indians, right? And we all know that the starting point of oral cancer, or what we call is OSCC, oral squamous cell carcinoma, it accounts for 97% of oral cancer cases. Right? It is a major thing, and uh, you know, a starting point obviously is the consumption of tobacco products, alcohol, nicotine, probably tobacco. It's a part of tobacco, right? And uh, to some extent, HPV virus inside our mouth, and to a larger extent, the microbes in your mouth, right? So how microbes in your mouth, they interact with the external factors, whether it is these consumption of these harmful carcinogenic potential products such as tobacco and alcohol and smoke, right? Uh, uh, right, and or probably certain foods which activates various pathways which led to production of certain microbial gene products, what we call is metabolites, right? And these gene products actually is as outcome of these gene products outcome of the expression of genes which leads to activation of certain proteins and enzymes which causes these products to start inflammatory cascades in the mouth, and so basically it's linked to epithelial cells along the oral cavity, right? And over the next few weeks, before we finally launch our you know oral cancer earliest detection test or platform, whatever way you call it, but we generally call it platform because it's actually not a test, so as to speak, right? We're going to discuss seven biological themes, right, around what causes oral cancer, basically OSCC, right? And these themes would include the antimicrobial pathways, the cancer-specific metabolism, detox mechanism, you know, volatile organic compound, volatile sulfur compound, right? Then pro-inflammatory pathways, and finally protein fermentation levels, right? So these are the seven uh, key biological mechanisms we cover, and each of the biological mechanism would have different microbial pathways. So we would be covering around 27 of such pathways, right? So we're combining these 27 pathways to determine, detect, or predict the onset of OSCC. So again, today we're going to start with volatile sulfur compounds, right? And it is one of the major reason for onset of oral cancer, right? So having said that, before, it is important to understand that uh, the most of the interventions today in terms of detecting or treating cancer is again at a subpar level or suboptimal level, because most of the interventions are linked to, you know, the invasive biopsy followed by histopathological evaluation. So probably then PET scan and a lot of these things, right? So basically, it is generally at a stage when you already have tumor developed, right? Uh, and this is, so as to speak, the advanced stage, right? And again, most of the interventions post this is restrictive chemotherapy and immunotherapy and a lot of these radiation therapies and surgeries, right? In case of oral cancer, it's mostly the surgery to remove the tumor, right? That's what is happening. And again, these are virtually, the success is very low, because again, uh, we are in the last stage, and we don't address the root cause. We just try to get rid of some tumor, right? And again, there are chances of reoccurrence, right? Reoccurrence of especially oral cancer is never addressed, right? So essentially, what we are going to do is we are going to uh, measure and quantify these pathways, which tells the epigenetic changes and the changes in the biochemistry before long years before the tumor starts developing, right? So that is what we call is the earliest stage, or even prediction, right? Detection, prediction, right? So essentially, uh, these are sort of indications which have never been measured by any of the detection tests also. So there are some tests which look at certain uh, enzymes, and then they tell, so that enzymatic activity is also an outcome. They only show up when you have tumor developed, right? So so we are going to talk of volatile sulfur compound, right? VSC, and it is a major issue. And before that, let us discuss some mechanics, right? So Sakshi, do you have any questions as of now?
I think you should continue because whatever
Yeah.
you were saying, we've already discussed a lot of these things, previously in our talks. So I'm pretty sure by now users they do know the kind of definitions that we have been talking about. So I think you should probably continue with the flow. I'll put up my questions after that.
Okay, sure. So how, let us understand what is a pathway first, right? So we talk of pathways, and a lot of people say, "Many समझ में आ रहा क्या है?" So, so again, a pathway is a combination of thousands of biochemical activity, enzymatic activities happening, right? And again, uh, what happens is, whenever there is an external trigger, whether you're eating something, whether you're taking supplements, the air you live, the environment you live, the sort of probiotics, prebiotics you're taking, supplements you're taking, tobacco products you're consuming, right? Uh, all these uh, have, what it happens, it activates certain microbes, and so this sense, is a sack of molecules, right? Everything, right? Even the food is a sack of molecules. So they sense it, and they express a particular gene, right? And when they express a particular gene, that lead to activation of particular enzyme or protein, which produces certain gene products or microbial gene products, or what we call as microbial derived metabolites, right? And these changes the physiology, these either activates immune system or give uh, nutrients, right? It activates immune system. There is an activation sort of human gene expression, and then there's a overexpression, underexpression, certain inflammatory, anti-inflammatory markers which causes disease. That's a typical thing, right? That's how a basic have, and this is what we call is the production. This is what we call is the pathways. We look at various biochemical activity happening inside the body, which triggers a particular gene product, right? So it's not something very linear that you're saying, okay, if I have this bacteria, I will have this. That doesn't work like that. So if bacteria would have different genes expression, some under expression, some other expression, different protein expression, right? And again, different these biochemical activities combine and become a pathway, right? So here we are going to discuss volatile sulfur compounds. Basically, from a oral cancer perspective, there are two pathways which we are going to discuss. One is, we'll start with probably I'll take it both together because they work together in tandem, right? So one is hydrogen sulfide, which is again an uh, oral microbiome actually metabolizing and processing foods, sulfur in foods, right? Or sulfur containing amino acids such as cysteine, homocysteine, taurine, right? Methionine, right? These are basically, so you will find these amino acids in a lot of animal protein in kidney beans, split beans, right? A lot of some nut seeds as well, right? Again, so it doesn't mean these are bad, but again, the which microbes, what is your microbial environment in the mouth which interact with these. So probably they could interact with the certain microbes, gram-negative microbes, or potential pathogenic or probably commensal who could do pathogenic activities, right? So these microbes metabolize or pre-process these sulfur in foods such as broccoli or other uh, cruciferous vegetables which are high in sulfur or probably sulfur containing amino acids, right? And uh, they could lead to expression of certain enzymes. We call it cystothionine beta kinase, which is again CBS, right? And again, this expression of this enzyme causes high production H2S, right? And in simple words, this overexpression also leads to another expression of enzyme called NAMPT, which is again, you must have heard of NAD supplements, right? So again, this is the enzyme which metabolizes NAD. And again, people who have a high risk of inflammatory activities causing cancer, they should not take NAD supplements. I think we have discussed this earlier also, right? Regarding NAD supplements, it's not good for everyone, right? People who are taking NMN and NAD supplements thinking it is going to improve
Yeah,
the
I
aging.
remember this from the NMN talk that when we were talking about supplements for these NMN aging, we
Yeah.
remember, I think it's from the talk of our age about aging.
Yeah. So
Yes.
what these things combine, so CBS, right, or NMPT, all these uh, interact with hydrogen sulfide. There's a positive feedback loop, and they leads to tumor survival, growth of tumor, right, and also cell differentiation. So increasing of tumor grade, so increase the size of tumor grades, right? And it leads to cell proliferation. Probably cell proliferation means the growth of cancer cells, right? The mutagenesis. So probably there is a so there is certain oncogene activation which causes onset and progression of cancer. So that is one of the story of hydrogen sulfide. So if your microbiome is producing H2S or methyl mercaptan, again there are two. One is H2S, one is CH3SH, which is again methyl mercaptan, which is a byproduct of microbes processing L-methionine, right? So you're consuming a lot of protein. People consume a lot of protein. So if you have certain protein supplements or having food which are high in L-methionine, probably you should look at how your microbiome is interacting with them, especially oral microbiome, because it could lead to methyl mercaptan, right? Proxy methyl mercaptan, which again have a lot of uh, inflammatory role to play, right? So we I just mentioned about the cell proliferation, glycolysis, right? The cell
Prashant,
growth, yeah.
I have a question because
Mhm.
since you've already used the word gene activation, you said that there's a certain gene activation for OSCC. So when we refer to the word gene activation, is it the DNA or the RNA? Because many
RNA.
people, yeah, I know. I'm raising this topic because many people who with limited knowledge in the industry, they think it's actually the DNA, but it's not.
So
So,
so let me tell you this thing here, right? That so our DNA is static, right? Just give an example. I think I've discussed this, but I'll briefly talk about this. That your brain, liver, heart, skin, everything is the same DNA because we are born with one cell zygote, right? And we have the same DNA, right? So again, there's every organ has a potential to become every tissue has a potential to become any of those organs, but doesn't know which organ is what function it performs, right? For instance, your liver and your brain have the same DNA, but they perform vastly different functions. So it's the activation, or I'll say the the gene's expression, right? Right. So essentially, when there's an external trigger, whether it's food, whether it is supplement, whether eating this tobacco and all these things, or environment, there's always DNA is copied into RNA, which is again, it converts gene sequence into particular protein expression of a particular protein, which perform a biological function, right? Or dysregulation biological function, which can be good or bad. Right? So again, what I mentioned by gene activation is the expression of the gene, right? Sorry if I've used that word if people have not understood. It is the activation of particular gene. It expresses a particular gene which activates a particular protein enzyme, which causes, for in this case, it microbes consuming, for instance, P. gingivalis consuming, these sulfur in other sulfur-containing amino acids or cruciferous vegetables, right? Probably it could lead to activation of a particular gene which produces H2S, right? So in simple words, I don't want to get into too much of technical detail, but produces H2S. H2S uh, is actually, outcome of expression of CBS, which is again an enzyme, or NAMPT, which is, so these three interact with each other, and it leads to positive feedback loop. It leads to cancer cell growth. It leads to tumor survival, tumor the differentiation, what is the growth of tumor, right? And uh, uh, right, and a lot of these. So this is one of the factors. And again, there are multiple other things how H2S or volatile sulfur compounds causes cancer. So one is, it suppresses something called SOD, right? Which is again an enzyme which is required to inhibit your oxidative stress. So it inhibits, when it inhibits SOD, it leads to production of free radical, it leads to oxidative stress, it leads to DNA damage, and alters the lipid and protein metabolism. So again, when it alters, it actually feeds the cancer cell, right? So again, that is again a harmful aspect of it. Uh, Second thing is, there are certain activation, certain signaling pathways, cellular signaling pathways such as, RAS and signal which leads to, which expresses a particular gene which leads to cell growth, right? So cancer cell growth happens, and there are certain bacterias. So H2S, one more thing, I think we discussed this for people who know there's another endotoxin region inside our body, what we call is lipopolysaccharides, LPS, right? And H2S actually increase the penetration, H2S and methyl mercaptan CH3 as such also increases the expression of LPS, right? It penetrates the mucosal lining and it leads to, mucosal permeability, which is again leads to carcinogenic potential and triggers cancer situation, right? So again, there are cellular pathways, there are mucosal intervality, there is the activation through certain expression of proteins, and then there is oxidative stress, which causes DNA damage, alters lipid and uh, protein metabolism, right? So essentially, these are factors. Besides, there are certain other more things I wanted to mention here is that collagen synthesis, it reduces the collagen synthesis. So H2S has a much wider role to play in terms of, the inflammation, right? So it suppresses your collagen synthesis. It can inhibit your wound healing, right? So this is again and side effect of consuming too much of this, too much of probably you don't know you are consuming foods which are sulfur containing amino
Sulfur
acids.
containing foods, I think then see sulfur is already, I think in one of our talks we had seen that white sugar is sulfurous in nature. Eggs have
Mhm.
sulfur, right? Carrots,
Cruciferous vegetable.
pumpkins, cruciferous
Cruciferous.
white
So
vegetables,
so
they all of
I
them have
doesn't, so for
sulfurous.
everyone it doesn't mean that you don't need to have to eat this. To
No, no,
understand your
but that
biology.
has to be optimized to a certain level because you know what
Mm,
happens when you eat a lot of that, it is going to create a problem for them, right? And
mm,
with the
mm.
progression of H2S, and I know that it starts with the mouth, but entirely the metabolization is happening inside the gut, so it is also going to cause the digestive problems. So early symptoms could be the rising of these problems
So
within
again, and
the
early
digestive system.
symptoms is these all are indications. So these two of the pathways are also what we use to predict halitosis. Again, halitosis is an early biomarker for periodontal disease. Again, those who have periodontal disease have a higher risk of OSCC. So probably if you have tooth loss, if you have a lot of gum disease indications. We have leaky gums. These are early indications for oral cancer as well, right? So, so essentially, these specifically methylmercaptan CH3SH, actually, it reduces the collagen synthesis by 40%, right? So it inhibits your wound healing. It also suppresses cytochrome, right, which is again required for oxidative phosphorylation. So basically, in simple words, the conversion of oxygen into ATP, right, which gives you energy. So it suppresses cytochrome, which is again it interferes with the oxidative proliferation cellular pathway, right? So this is something which is really important to understand, that this is one of the pathway. Doesn't mean that this is the only reason for OSCC or oral cancer, right? So volatile sulfur compounds, your microbe consuming a lot of foods which are high in sulfur or methionine or cysteine or homocysteine or taurine, right? These all are amino acids, or maybe a lot of sulfur, right? Or methionine, right? So again, methionine is a sulfur containing. It also produces methylmercaptan, right? So essentially, these uh, bum, microbial gene products, what we call as metabolites, are uh, can trigger our outcome of expression of certain proteins, enzymes, which causes the cancer cell growth, and it leads to oncogene activation and a lot of these factors which contributes to cancer. So it's not something which happens overnight. So these things you are doing, you don't know, you knowingly and knowingly you are doing, because these are asymptomatic, right? After some time, you do have that bad breath, right? That is an early indication for it. But again, this is one of the early biomarkers which we look at, right? From an uh, prediction onset of OSCC oral cancer, right? And again, these are, I'm not sure how many cancer testing companies or doctors look at halitosis or bad breath, or look at H2S production, look at methylmercaptan production. Is again, so volatile sulfur compounds. Again, this is something which impacts your energy metabolism, impacts your lipid protein metabolism. It causes oxidative stress. It causes DNA damage. It causes mucosal permeability. So it could lead to overexpression of LPS, overpenetration of LPS into the gingival tissues, right? So it is all the epithelium around your oral cavity, the epithelial cell lining on the oral cavity, right? So essentially, these all things are happening at a molecular and cellular level, which are not reflected in any of the evaluation or histopathological evaluation, or even if your biopsy, right? Your
Yeah.
your your invasive biopsy, right? So what we do is, what we are going to do probably is going to launch it on the eve of Independence Day, right? But uh, uh, what exactly we look at is, we take your non-invasive saliva sample, right? We collect your molecules, RNA molecules, we analyze them and identify which microbe, which gene it came from. We combine them into various pathways, and again, various pathways are measured and quantify. Different pathways are further combined into biological mechanism, which is again calculated mathematically to tell you whether you have this oral cancer or not. It is at even stage one, right? Even before that, right? So essentially, even when you have not developed, so essentially you know this, and then you could actually. And then we also, unlike a lot of testing detecting companies, we also provide you intervention in terms of nutrition, which could eliminate these molecules well before the disease manifests, right? Well before the cancer develops. So this is one of the biological mechanisms which we discussed today, right? And
Yeah, I think going forward we'll be discussing more pathways which leads to such OSCC related issues, right?
yeah,
And I think with this we have come to the conclusion of this talk. So
yeah.
Sushant, any last words would you like to add here?
Yeah, so basically, these are asymptomatic, which doesn't come up. No test will identify them. Uh, for cancer to develop and show up in your biopsy or PET scan, you have to wait for at least 5-10 years to develop. Right? So essentially, that is our objective. There's a huge gap in detecting these things at a very early stage, and that's what we are doing. Right. So essentially, today we discussed about the volatile sulfur compounds. Next talk we would be doing on the cell specific energy metabolism, how the cellular molecular interaction or dysregulation leads to how we are feeding our cancer cells instead of feeding our mitochondria to produce ATP to give us energy, right? So we're going to discuss that thing also. So I've kept it very, very simple, not too much of technical, but again, this is really, really complex thing, and that's why you really need to understand it. So that's it. We'll come up next week with the next talk. Probably we'll doing it every week, sort of, discussing this. And if anyone has questions, please do reach out to us. We would be sharing our email in the description box. So probably you could reach out to us. And if you have any questions, if you want to know more, or probably you want to understand your risk for other diseases such as type 2 diabetes, cardiovascular disease, chronic kidney disease, or even halitosis, reach out to us. These are the 4 diseases we predict right now based on gut and oral microbiome interventions, right? So that's it for today. Yeah.
Thank you, Sushant. Thank you.
Thank you.